This is Medicine Jerk. Today we are going to discuss one of the most important topic, that is Warburg effect. Okay, Warburg effect. And this Warburg effect is useful in one of the most important radiological investigation like PET scan. Okay, na? PET scan. So today we are going to discuss about the Warburg effect. Before discussing it, we should know what is the fate of glucose within the normal cell or the normal glucose fate. Okay, na? Normal glucose fate. Assume this is a normal cell. This is a normal cell, and of course normal cell takes up the glucose. Okay, na? And this taken glucose may enters into the two pathways based on presence of oxygen or the absence of oxygen. If the oxygen is present, it forms pyruvic acid. If the oxygen is absent, it forms the lactic acid, which is called as anaerobic glycolysis. And the formed pyruvic acid will enter into the mitochondria. That means it enters into the Krebs cycle. It enters into the Krebs cycle. By the help of the Krebs cycle, the cell forms lots of ATPs. Lots of ATPs which are useful in the normal metabolism. Okay, na? This is a normal glucose fate. Okay, let's see. Let's assume this one is a tumor cell. Okay, na? This one is a tumor cell. Okay. So what happens within the tumor cell? Tumor cell will also take the glucose, but the uptake will be maximum. Okay, na? Uptake will be maximum. Okay, na? And whether the oxygen is present or absent, within the tumor cell, glucose will forms lactic acid, and this formed lactic acid will enter into the mitochondria. Okay, na? After enter the mitochondria, it forms three products. Okay, na? Three products. One is lipids. First one is the lipids, and second one is the nucleotides. Well, the third one is proteins. Okay, proteins. All these three together forms the building blocks of the proteins. Building blocks of the tumor cells. All these three together forms building blocks of the tumor cells. Okay, na? So that the tumor cells can populate very easily. Okay. So the glucose fate within the tumor cell, what we have discussed till now is nothing but the Warburg effect. It's nothing but the Warburg effect. Okay, na? It is Warburg effect. In the simple terms, here the glucose is converted into the lactic acid, whether the O2 is present or absent. Okay, na? So all the cells actually has the O2. Okay, na? So glycolysis is also going. So what type of glycolysis it is? It is aerobic glycolysis. And basically the tumor cells will take the glucose in the maximum quantity. So this can be termed as glucose hunger. So glucose hunger is seen in tumor cells. So now this Warburg effect is used in one of the most important radiological investigation, that is PET scan. Okay, na? PET scan. What is the full form of the PET scan? It is positron emission. It is positron emission tomography. And the principle which is used in the PET scan is annihilation coincidence. Okay, the principle which is used in the PET scan is annihilation coincidence. Okay, annihilation coincidence. So we'll take the patient into the radiological investigation room, and we'll inject 18 fluorodeoxyglucose. Okay, 18 fluorodeoxyglucose. If the patient has the tumor cells in his body, we can is present tumor cells in the body, so it will take up the lots of lots of 18-FDG, lots of F18 glucose or 18-FDG, 18-FDG. In this way we can easily visualize or locate the tumor cells. So this helps in the early diagnosis of metastasis. And you also, everybody knows that metastasis is a late feature of cancers. Okay? This is Warburg effect. Thank you. Signing off.